Hello everyone, this is another presentation. This time we are going to be talking about Oncovin, also known as vincristine, and this is a chemotherapy drug that is used to treat different kinds of cancers. Just a little background on the drug. Oncovin is also known as vincristine sulfate, which is an anti-cancer chemotherapy drug. It's used to treat cancers such as acute leukemia, Hodgkin's and non-Hodgkin's lymphoma, neuroblastoma, rhabdomyosarcoma, Ewing sarcoma, Wilms tumor, multiple myeloma, chronic leukemias, thyroid cancer, brain tumors, and it can also be used to treat some blood disorders. It's administered via IV injection, and it is a vesicant, which means that it is a chemical that can cause extensive damage and blistering if it escapes from the vein, so they have to be careful when they're injecting it to ensure that it does not come out of the vein and start causing swelling and irritation in the surrounding tissues. The other side effects that may occur from administering this medication, not a lot of people experience these side effects, but these are the ones that were listed. They can be hair loss, constipation, low blood counts, abdominal cramps, weight loss, nausea and vomiting, mouth sores, diarrhea, loss of appetite, taste changes, and peripheral neuropathy. Sorry. So the way that tubulin works with the oncovin and cell division is tubulin is a protein that polymerizes into long chains or filaments that form microtubules. These hollow fibers serve as a skeletal system for the living cells. The microtubules are what aid in mitosis to help regulate intracellular transport because they are able to shift through various formations. The mitotic spindle, which are composed of a bipolar array of microtubules, aid in the movement and segregation of chromosomes during mitosis. The tubulin code has recently emerged in studies and has been shown to add as an extra layer of regulation governing the microtubule function. And the way that microtubules work is they invade the nuclear space and attach to chromosomes at the kinetochore. Upon attachment to the kinetochores, microtubules then promote chromosome congression to the metaphase plate and later segregate the sister chromatids to opposite poles during anaphase. So oncovin or vincristine is a natural alkaloid isolated from the leaves of the Madagascar periwinkle, which is a flower. The way this drug works is to inhibit microtubule dynamics that normally cause mitotic arrest and cell death. It also works as a microtubule destabilizing agent by stimulating mitotic spindle destruction and microtubule depolymerization at high concentrations. At lower concentrations, it can block mitotic progression, and unlike taxanes, vincristine can bind both soluble and microtubule-associated tubulin. And vincristine rapidly and reversibly binds to soluble tubulin to stabilize the kinetics of the microtubules. This can increase the affinity of tubulin by inducing the conformational changes of tubulin. After that, vincristine then binds to beta-tubulin subunits at the positive end of microtubules at a region called the vinca binding domain. The rate of microtubule dynamics, which is lengthening and shortening, is decreased from the binding between vincristine and soluble tubulin, and it increases the duration of attenuated state of microtubules. This means that the assembly of the mitotic spindle can be prevented, and the tension at the chromosome's kinetochore is reduced. The chromosomes then cannot progress to the spindle equator at the spindle poles. The cells then can enter mitotic arrest because progression from metaphase to anaphase has been blocked. The cells can then undergo one of two options. The tetraploid cell can undergo unequal cell division, which produces aneuploid daughter cells, or it can exit the cell cycle without undergoing cell division, and this is called mitotic slippage or adaptation. The cells can then continue to progress through the cell cycle as tetraploid cells, or they can exit the G1 phase and undergo apoptosis, which is cell death, or they can escape to G1 and undergo apoptosis or cell death during interphase. The cell may also die during mitotic arrest, or mitotic catastrophe can occur and also cause cell death. The cells have developed some resistance against vinca alkaloids by drug efflux, which is mediated by a number. And then on this last page that I have here is the molecular structure of oncovin. Let me just try and make it a little bit bigger. It's a pretty big molecule, so you can just kind of see that. And then this is the periwinkle flower that the Oncoven is made from. So I just wanted to include that so you can kind of see where it basically originates from. And then over here, we have a picture of the tubulin and how the vinca alkaloids prevent the assembly of the microtubules. And then the taxanes prevent microtubule disassembly down at the other end. So then we have the alpha tubulin, which is shown by the blue dot, and then the beta tubulin is shown by the orange dot. So you can just kind of see how the Oncoven prevents the microtubules from assembling. And then this just shows what a microtubule looks like and what it's made of, which is the alpha tubulin here and the beta tubulin. So you can kind of see those are just wrapped together to create a microtubule. And then on the next slide, I have my sources that I use to find the information on this. So you can look those up and see if you can follow along with that and see anything else in those. But this is just to kind of guide you along if you need more information on Oncoven. Thank you. I hope you liked my presentation, and have a good day.